let's start the session. So we have just 3 talks, 1 half an hour and 2 15 minutes. And the first speaker is Sandeep Choubey from IMSC Madras.
Hi, I'm Sandeep. First I would like to thank Dimple, Mithun and also Ranjith for organizing this fantastic conference and also inviting me to present some of our work that we have been recently doing. So I'll start, of course, as you can see from my slides, I'm going to talk about something related to genome compaction. Again, there is a slide, of course, that I have to start with, as all of you know. So, of course, all this complex stuff, I don't have to say much about this, but one important thing is that proper genome organization is essential for all domains of life, be it eukaryotes or archaea or bacteria and so on. So, of course, the biophysical mechanisms are quite distinct between these groups, and of course, we try to understand how it's compacted. That's the general question. Now, of course, all of it really boils down to this question of how a protein interacts with DNA, because you see, all these different processes we have been hearing about, CTCF, cohesin, and so on. Essentially, it's all about protein-DNA interaction, really at that level at the molecular level. So, probing them often inside cells is challenging, and again, you have many factors. As many of us said, that the systems are inherently multi-component, active, and so on. So really doing controlled stuff is very difficult. So testing any models or theory is extremely challenging. But on the other hand, you can take an alternative approach of really probing DNA-protein interaction through in vitro studies. And often the setups that are employed are the following. So Mahipal already talked about this, that you have a cover slip-based assay. You tethered the 2 ends of a DNA, usually the lambda DNA, but of course you can use other DNA constructs as well, and then you flow in some proteins. And this is a beautiful experiment from a group in Dresden where they looked at FOXA1 interacting with the DNA, and you can see that they form these like clusters along with the DNA. And the usual picture, the way they kind of try to explain it is that these proteins like each other, and they also like the DNA, so they're together forming some sort of a structure. And of course, they call it a condensate. So the other kind of setup that people employ, this is from Geeta Narlikar's group, is that you have an optical tweezer assay. You again tether the 2 ends of a polymer using biotin beads, and then you observe how proteins interact with DNA. Right? Now, this is kind of a schematic that describes what's happening here, that you have protein and DNA together, that's forming some sort of a co-condensates. In other words, it's not just proteins are condensing alone, but also the DNA is in a globular state. So the DNA is really getting compacted by the protein, and they're together participating in this process. It's not like the DNA is really a passive substrate. Usually the end-to-end distance is kind of one thing that you can tune in experiments and so on. So one of the key goals for us is to really understand these kind of data. We want to build a modeling framework to understand exactly these kind of experiments. And there are many such experiments that are being done now, so we're really interested in that. And the other thing that we want to think about is the role of the DNA sequence. Of course, we know DNA sequences, the DNA is heterogeneous, and each protein sees the different parts of the DNA with different affinity, like it binds to different parts of the DNA with different affinity, and so we want to understand the role of DNA sequence. Right, so again, it's a very simple minimal model, kind of an abstract representation of a protein and DNA. So the DNA is essentially represented by a bid, like a bid spring, where each bid is of the size of a footprint of a protein of interest. And the protein is also a spherical bead of the same size as a bead here on the DNA. And of course, you can take different sizes of DNA. We took a size of 5 kb, and assuming that each protein occupies 10 base pairs, basically you have a 500 monomer long polymer, and you kind of study that to begin with. And the usual, the interaction energies that we consider are the following, that we basically say that, okay, they are of similar magnitude of 2 kT, basically. right? Of course, you can vary these and study the same system, and you get interesting features. But we start with this. So this is the kind of simplest thing one can initially think about is, So you have, if you take a bunch of proteins inside a box, which interact amongst themselves without the DNA, at low concentration, you just see a homogeneous solution. And as you increase the concentration, you will see that at a high concentration, they form this kind of a droplet, right? Now, you go to the low concentration one, and you add DNA, which is tethered at two ends. And this is exactly what happens. So essentially, you are condensing the proteins, even at this concentration, where you're not supposed to see any droplets. So essentially, the DNA and protein together are forming these structures at a much lower concentration. And which concentration you can go down to depends on the interaction energies and so on. Now, this has been, this is something that has been studied in the last few years, in particular from Daniel Jost's group and other, like Marindoso and also Helmut Schitzel from Dresden, and they have seen this phenomena. Now, this is, as I said, we call it the protein-DNA co-condensate. You can now start quantifying various things. The first thing we are kind of thinking about, okay, what happens to the number of condensates? Like, as you would expect, something like this, we had initially, you saw that there are two condensates that form at different positions, but then they merge. Either you merge and become one, or like the smaller condensates lose material and the bigger ones grow. Now that is known as oswald ripening, and that just happens because of the difference in Laplace pressure, so the bigger ones tend to grow. And it's like monopoly in some sense. So then you can look at their position and see the size as you change concentration. This feature is robust, you always see one condensate. Okay, now we said, okay, let's actually add some heterogeneity, because we want to understand actual DNA sequence heterogeneity and its effect, we first take a simpler version of it, where we say at the middle of the DNA, we introduce a high affinity region, and there is a low affinity region in the flanking parts. Then you again simulate it. What you see is the following. Of course, as they form at different locations, they come and merge. And as you would expect, they form in the middle, which is not really surprising. So really the sequence is playing a role, which is not really surprising, but something that we want to test anyway. And again, you see, if you run it for different concentrations, they form in the middle and so on. Now, the other thing that we like to think about then is, what about the forces? Because you see, the moment you have something like this kind of a structure, they are actually pulling the other bare DNA into the structure, because the proteins like the DNA, right? It wants to bind to various parts of the DNA, so it likes to pull this part in. So there is some force that is getting generated, and of course, as you can imagine, something that we are taught also in high school physics is like the concept of capillary force. It's essentially like a capillary force. Now, if you look at the force, as you tune the end-to-end distance, so of course you have the DNA tethered at the two ends, you can change the distance, and that's the normalized distance, which is basically normalized by the contour length of the DNA, like the actual size of it, and then you do not see much of a difference for the homogeneous DNA. Now, of course, as we know, like as Pauli said at some point that God made the bulk, the surface was invented by the devil, right? The moment you have surfaces or interfaces, all sorts of like interesting physics emerges. And now you go to the heterogeneous DNA, just the one that I saw. As you see, for up to some end-to-end distance, 2.5, you see that it's almost constant, and then there is a sudden jump. And the jump is simply because initially, the condensate was large, for instance here, and you see the yellow beads at the interface, right? So the proteins are interacting weakly to these interfacial DNA sites. And because of that, it really doesn't pull them with that much force compared to where you have a higher end-to-end distance, because the condensate has become smaller, and the red beads have actually come to the interface. Now suddenly there is a jump in force, right? And you can see the forces can go from 2.5 piconewton to 5, like it's almost twice. And of course it depends on the affinity difference we chose, which is 0.5 in this case, 0.5 kT, which is within the range of things that we observe in actual biological systems. So clearly heterogeneity has some interesting effect on the forces that like this kind of co-condensate imparts on the rest of the DNA. Now we took an actual sequence, which is a partial lambda DNA sequence. In other words, what we did is that we took the whole 48 kb long lambda sequence, and a part of it we employed here. And again, as you can see, if you look at the AT content, for instance, it's very noisy. It's all over the place. It's barely you can see any signature, right? Like where the proteins will bind and so on. Now, what we did is that we simply said that there are many proteins that bind to AT or GC-rich regions. We just simply said that, look, we'll say that the beads, essentially based on this, we take 10 bases and see what fraction of it contains AT rich, or what is the AT richness of that 10 base pair sequence, and based on that we assign some energy. And this energy is linearly assigned as a function of the fraction of AT content. So once you do that, you again see something quite interesting, which is that you run the simulation for a really long time. You can actually see that there is arrested coarsening. You see two condensates that form at two different regions, but they are not merging anymore. And the reason we understand so far, this is something that is still work in progress, but so far what we understand is that, of course, coarsening is happening because of Laplace pressure and, the usual lossful ripening thing. But now, there is another force at play, which is that these condensates are actually applying force on the DNA, because every condensate wants to grab more of the DNA, right? This one is trying to grab some DNA from here, and this one is, so there is some sort of a tug of war going on between them. And once the tug of war goes, say that somehow this smaller condensate gets an interface which has a higher affinity than this one. So it can actually negate the internal Laplace pressure that it's feeling. Of course, it's a complex system because you have a really multi-component mixture of these monomers and proteins and so on. But that's how we think, what we think is really going on. Now, through that, then you can really see these multiple condensates. And it also has an interesting impact on simply as I said, if you look at the lambda sequence, it's very noisy, but when you change concentration, you can see these really sharp jumps in forces, really nonlinear jumps, and it all has to do with the interfacial DNA. Like, say that a condensate is smaller initially, when you have a lower concentration, you increase the concentration, the condensate grows a tiny bit larger, then it grabs a bit of more of the DNA, and so the interface changes, and say that the binding affinity, like the moment interface changes, some more monomers come in, and the monomers that come now has slightly higher affinity. That can have huge impact on this. Now, for instance, in this case, if you want to understand, you see this is for lambda DNA, in which case you will see something like this. And then this is the other picture. And if you look at the affinity of the interfacial DNA, of course, it's fluctuating. So you have a whole range of different monomers that are coming and going. So we look at the overall the average energy of these. And you see that there is a tiny bit of difference of about 0.3 kT. And that leads to this sharp change in forces. Right? So in other words, really base pair level stuff is impacting the amount of force that you are imparting on kilobase pair long DNA. Right? So very local changes can have really large scale effects. Okay, so then we wanted to see, okay, based on whatever we have learned so far, can we actually shed light on some of the existing data sets? Right? And so the first thing that we did is to look at this particular protein DPS, which forms a complex with DNA. And also DPS is known to phase separate, and it's something that people have seen in vitro studies, something also Sneha talked about on the first day. So you see, DPS seems to always form a single condensate. And you look at a range of different experiments, they always do, like you can change concentration and so on, it always tends to form one condensate. And it's known that DPS binds DNA in a non-specific manner. So they don't really have well-known consensus sequence or anything. So it's kind of consistent with what we expected. Then we thought, okay, let's look at a protein where you really have a well-known sequence effect. Now, this is SOX2, and we know that SOX2 has a well-defined position with matrix and so on. If you look at it, and I mean, this is I'm just showing one representative image. There are many, many such images, and you can statistically see that there are always more than one condensate. Of course, whether the system has equilibrated or not is always an issue. That's an issue with the experiments, also with simulation. That is there. But if you see it for a long enough time, you do see that there are these stable structures that have formed multiple structures, basically, which is consistent with. So it's of course that essentially tells you that rules out or falsifies the homogeneous DNA model. So that's clearly ruled out. So clearly there is some sort of heterogeneity, which is, but of course it's not surprising in that sense. But then we look at some other protein, which is HP1. And I'm sure many of you have thought about HP1 and so on, or have done experiments. So this was this data from Gita Narlikar's group, and they kind of saw two condensates continuously for very different, in very different experimental conditions, rather than one, which is actually consistent with a heterogeneous DNA picture. And you see, you see this is actually contradictory to what people think, because people think that it doesn't bind in a sequence-dependent manner. It has a sequence non-specific binding to DNA. But of course, I was talking to Mahi later, and he was saying that it's potentially possible that it does have, and they also have some preliminary observations that seem to suggest that. Okay, so then we go on to talk about a collaboration that we have, running collaboration that we have with Mahi. Mahi's lab. So this is Tejas, who was an undergrad, now moved for his PhD position to Netherlands, and also Prakshi, who is in the crowd, and of course Mahi. They have been looking at this protein, which is known as LSR2. It's a nuclear associated protein, and it also there are AFM images where people have seen that if you add DNA and protein together, you form these structures, right? We want to understand, again, the biophysical properties of these and how they actually really form and so on. So this is basically, these are the two questions. What are the physical principles of LSR2 binding to DNA? One. Second is, again, what is the role of DNA sequence? Now, which is this data Mahipal already showed, but I wanted to show it anyway, is that if you increase the concentration of LSR2 in an in vitro solution, you see that the number goes up, right? There are condensates that form at a concentration of about 10 micromolar. Now you add DNA to the solution, which is basically 1.8 kb long DNA, and what you see is that condensate starts to appear like these tiny structures start to appear at even a higher concentration, like even at 0.5 micromolar or definitely at 5 micromolar. So clearly DNA is reducing the saturation concentration for these proteins. Okay. Now the experimental setup, of course, is this cover slip-based assay, which Mahi talked about, and this is what you observe. On the left-hand side, you can see that you have the DNA, and then some are single tethered or they're breaking bonds, but otherwise you see these structures that form, and this is lambda DNA again. And this is the corresponding protein channel if you look at it. So one important thing is that if you look at the AT content, again, it's at a 10 base level, it's very noisy. You coarse grain it because you have the resolution issue in experiments, because you cannot measure arbitrarily with an arbitrary precision. You have each pixel corresponds to about 500 base pairs, and that's why you coarse grain over it. And this is what you kind of get. That's the coarse grain landscape. Now, again, we do the simulations with the actual rugged landscape, not the coarse grain one. And this is what you see in the simulations. This is where they form in the middle, tend to. Of course, you do many simulations. Sometimes they also form stochastically at different locations, but you can run it for many and then look at the average behavior. And this is what you see in experiments, right? And this is again one representative movie. So as you can see, that this is what the model predicts, and this is what kind of experimentally you see. And of course, it's one representative image, we average over them. So this is what you get from the model. You can say which part of the DNA is inside the condensate and not, and you can look at some sort of a distribution of this along, like the probability distribution of that along a distance, along the distance of the DNA. And this is what you see in experiments. And this is the underlying sequence. As you can see, it's the coarse-grained sequence that the experimental as well as simulation data seem to match really well with. Now, what does it really tell us? There is something interesting that happens, is that proteins are binding at a single molecule level, right? So each protein is seeing only 10 base pairs. But because of this collective behavior, the way they actually search a particular part of the DNA is not through individually strong sequences. Because if I have a single molecule picture, if a protein comes, it will tend to bind more often to these like high affinity regions. You can bind here, and so on, right? But you always tend to form co-condensates at regions which have on an average a higher binding affinity. So it's the average that matters, not the individual affinity, right? So this is what the real difference between a single molecule binding picture and this kind of collective binding picture has. And something, again, I think Leelavati was talking about during, all the bioinformatic analysis that they do, something like this seems to be there also. Okay, so I'll just conclude. Basically, of course, as we learned so far, is that the homogeneous DNA always leads to one condensate through ripening. And then heterogeneous DNA can sustain multiple condensates for, these are quite stable structures. At least within the simulation timescale, we saw that they really remain for a long time. And then that can lead to really kind of interesting kind of effects on the level of capillary forces that get employed. And again, LSR2 seems to compact DNA through a sequence-dependent co-condensation manner. With that, I would like to acknowledge Rohit Singh, who is a PhD student. He did the simulations along with Pinaki, who is a postdoc. He's also here, and also he has a poster. Tejas is an undergrad, and Prakshi is a PhD student, as I said, and of course Mahi. And I would also like to thank the funding agencies, the DBT for Ramalingaswamy Fellowship, ICMR for another grant that we got recently, and of course the DAE Computational Biology Group. So the other thing that I wanted to briefly mention, that we have a PhD program at IMSC that started about 10 years back. So we look for students from different background, of course, physics, computer science, mathematics, and also to biology and so on, which is a really nice mix of set of people that we have, and we like to think about biological problems from different approaches. So of course, please spread the word, and you are always welcome to visit us and so on. Okay, thanks a lot for listening. Happy to take questions.
Yeah, okay, so there are quite some time for questions. Okay, I'll start with you and then you.
So thank you for your nice talk. So more AT-rich sequence looks more condensed, right? And then I'm wondering what kind of factor can contribute to these things? For example, DNA property, AT-rich DNA property can contribute to the facilitation of the condensate, or protein binding property to the AT-rich sequence.
So far what we understand is that what really matters is not the individual sequence. What matters is the collection of sequences.
Okay.
So if you are surrounded by really even moderately high AT sequences, but if the average AT content of a stretch is higher than the other parts of the DNA, then you tend to form co-condensate there.
Okay.
So I can imagine a situation where they are all in some intermediate level, and there are very high and low values, but then they may not form any co-condensate because simply the average level is low. So it's really the average part that really matters.
Okay. So that means very likely to be DNA property of by sequencing?
Yes,
Oh,
yes.
Thank you.
There's one more thing.
Yes.
Here, all the phenomena you are seeing is very energy dominated. Means actual will be much longer. You have only 500 beats. If you either make the chain much longer or reduce
the energy gap, yes.
The entropy will play a
Of
role,
course,
and
yes,
that will
yes.
break up into
Absolutely,
multiple
absolutely.
droplets.
That is there.
I didn't ask a question. I just commented. Okay, so I said that what he has been looking at is 500 monomers of the DNA, and the energy of interaction between proteins is 2 kb and so on. If we took longer chains where entropy of the polymer played a greater role, or alternatively reduce the value of energy of interaction, that could lead to smaller droplets forming, as was seen in experiments. So at the moment, his thing is more energy driven. So entropy, yeah, we can talk later. Madan.
Yeah, it's related to this, but not necessarily entropy, because it's known that if you take drops of water and put them on a spider web, they break up into very tiny droplets, and that has to do with more the surface tension between the liquid and air versus the liquid and the fiber. So again, if you can vary the surface tension, do you see changes? You're taking the energy scales to be the same, so I guess you're...
So we are playing with that. You were right.
Yeah,
you are absolutely right that it really depends on the scale. In particular, the sequence, if you see the energy scale, like the difference. Now you can keep reducing it. The moment you reduce it too much, of course, you see coalescence again. So you have to, you need a rugged landscape to really sustain them.
So the related question is that, is there a renormalized surface tension is there a reduction in surface tension, effective surface tension, when this process is happening? And
yes, I think so. I mean, that's kind of our hunch, that what is really happening is, because you see, the organization of the droplets is also very important. So of course, say that there are monomers which are not as strongly attracted to the proteins, they tend to go to the surface. On the other hand, if they really like the DNA, they will be inside. So the organization becomes important. And I mean, we have also been thinking about looking at the internal stresses and seeing how that changes over, I mean, based on it's just the parameter space is too large, because as you can see, there are too many things to change, and of course, running these simulations take forever. But I think those are all important features.
So on that note, oh, sorry, was there someone in front of you? No, you go.
So it was a short question, in the video where you did not have the DNA, the coalescence that you form the large droplet. I was wondering, why didn't you see at least the initial stage multiple small droplets? Because it seemed like you suddenly have the droplet.
Oh, no. I mean, these are really made faster. The video says that. Of course, you will form multiple. Yeah, absolutely.
So on that note, so in the simulations, you have thousands of molecules, and so as you go down to say 10, what happens to the surface tension?
Yes, then you can't really define a surface tension. What is a surface even then, right? I mean, that is a bit tricky, because really surface tension is, I mean, again, something we think of in terms of a continuum. But here, defining a surface itself is problematic. So, you can think of it, but with 10 molecules or 5 molecules, that might be tricky. Anyway, go ahead.
What would the equivalent measure be? Is that just the entropic trade-off then?
I don't really have an answer to it. Of course, you still are exposing, because I mean, the proteins would still like to be in contact with each other. So, but then the surface, I don't know. I really haven't thought about it much. That's a good question.
So, did you account for the rigidity of the DNA in your model?
Yes.
And so, I mean, this could depend also on the sequence. So, if you have heterogeneity in the bending energy,
yes, I mean, that's something we want to study, but we haven't so far. Again, I mean, actually, there are a lot of things that we can do, as you can imagine. But of course, we have data in mind that we are trying to see how much we can explain with this simple kind of model. But this is something the PhD students keeps asking me, oh, I should do this, and I keep stopping him, don't do this yet, because you don't even understand this system properly. So anyway, yes, thank you. That's. Yes, absolutely. All these things, but we wanted to see what is the simplest thing that can give rise to the fact. And it was not very evident to us in the beginning, before doing the simulations, that it can happen. I thought that, ripening would happen. But of course, in experiments, you saw that there are multiple of these structures. So we wanted to see how to actually, if we can explain it from a sequence perspective. Yes, I think so. I mean, maybe that's a potential, absolutely. That's a potential mechanism as well that can play a role, absolutely.
Okay, so let's thank Sandeep again.
Thanks a lot. Thank you very much.